The next set of analyses that I'm going to walk you through deal with the structure of the protein. And we do already have some information about that. As you can see here, our mutation deals with a series of amino acids that have a function. But what we're going to specifically look at in this screencast is three-dimensional structure at the atomic resolution. Now, there are two major sites we can go to. One is under NCBI, its structure database. The other is the protein data bank. This one is US based, and the PDB, the protein database, is European. We're going to stick with this one because the software is a little bit easier to use. So if we type in our gene name, we may actually get structures immediately, and here we go. According to this database, there are 7 known high-quality structures. These are built by X-ray crystal analysis or NMR, and we can take a look at all 7 or pick one. Now, what I would suggest you do is pick one that applies to what you're studying. Now, obviously, we're looking for cardiac troponin. Both these seem to deal with that. There are going to be other structures that might show up in your search that may not be directly related, so be very skeptical about these and don't just click on the first link. So what I'm going to do is pick this one right here, actually, just because I've already looked at it. So if we go to the link, it will bring up a structure page. This does have a short little thumbnail of an image here, but this is not a manipulable structure. The reason I bring this page to you is for 2 reasons. One is to show that there is a reference here which you absolutely must list. These are the researchers who did the X-ray crystallography analysis to determine the structure. Now, if you want to actually view and manipulate the structure, click here on View Structure. Your browser will download a file, and you'll need an application known as CN3D. CN3D, yeah, it's kind of a joke, I suppose. But literally the letters CN and then 3D. You can download it from an NCBI page. If you open it up, the file, in that particular application, it'll look something like this. A few things that I will highlight in terms of what you can do. Up at the top, which is off screen right now, you can choose different modes of looking at this. Under Style, you can choose different views of this, either as tubes or as wires. The most accurate in terms of function is space fill, so I'll click that right here. We can obviously move this around, as you can see here, just by clicking through. Other ways of manipulating are possible as well. You can translate the view up and down, but I'm just going to stick to rotation for now. There are 2 different proteins shown here. If we click in the background again, A and B are different colors. Blue for B is troponin I, while the other one is troponin C. So this particular structure will be especially useful of seeing where these 2 proteins interact. Now, other things that you can do is change coloring. These are currently colored by molecule, so one protein and the other each have a different color. However, we can change that to show, say, the hydrophobicity of different amino acids, or we can go to the charge of different amino acids. That may or may not be interesting or useful to you once you find the mutation. I'm going to go back to just the 2 molecules. Next, down below here, we actually have the amino acids that are visible here. Keep in mind that not the entire protein is visible in the structure. Oftentimes only fragments are used for the crystal, or only certain fragments crystallize well in a stable manner to be viewed. So we do not have a complete troponin I or complete troponin C here. So you're going to have to be look for the region that was mutated in your case. So let's go back to our sample sequence, and this is the area that was changed. You can sort of get a feel for that residum qual, I suppose you could call it that. And if we go back to the structure, Look and see if it's visible here, and it is not. So this would not be an appropriate structure to use. So if we go back to the structure list, let's pick another one. Download this structure. I will open this up, and let's see if we can find it here. So again, we're looking for troponin I in a particular sequence and looking for its function. Troponin I is C in brown here. If we go back to the structure in CN3D, this is, again, protein C. Here it is. We're looking for the sequence of amino acids starting with an RIS, and there it is. I'll highlight the entire thing. 7, 8, 9, 10, 11, 12. There's the region. And as I change the coloration, you'll see why this program is especially useful. So if we go again to spacefill and molecule, as I choose different amino acids here to highlight, they will actually highlight in yellow on the molecule. So if we go back to this structure, we can see exactly where this location is and what it might be useful for in terms of protein-protein interactions or, say, protein and substrate interactions. In this case, we can see this area is directly interacting with another troponin molecule, in this case, troponin C in purple. So what you'll want to do is use that information for your analysis. Note, you can also look up these proteins in the PDB. That would require a different program called JSMOL, which I've provided you links to as well. But again, what I would suggest is using the NCBI database and the program CN3D.